All right, today we're going to dive into a process that is no exaggeration at the very heart of modern molecular biology. It's called nucleic acid hybridization, and it's the secret to how scientists can find a single genetic needle in a truly massive haystack. So just think about this for a second. You've got one lonely, single strand of DNA floating around in a cell, or maybe a test tube. How in the world does it find its one and only perfect match in that incredibly crowded, chaotic environment? Well, the answer is just an incredibly elegant process.
Okay, so to really get a grip on this, we're going to use a pretty simple, but honestly, a really powerful analogy. Let's think of this whole thing as a molecular zipper.
So what is hybridization really? At its core, it's basically molecular matchmaking. It's this natural, built-in tendency for a single strand of DNA or RNA to find and lock on to another single strand that has the exact opposite, or complementary, sequence.
And this right here is the secret sauce. It's all about what we call Watson-Crick base pairing. It's a fundamental rule of life. A, adenine, always pairs up with T, thymine, using 2 hydrogen bonds. And G, guanine, always pairs with C, cytosine, using 3, which are just a little bit stronger. This super-specific, predictable attraction is what drives the entire process.
Right, so let's get back to our zipper. The individual bases, your A's, T's, C's, and G's, they're just like the teeth on a zipper. They will only, and I mean only, lock in with their one correct partner on the other side. And it turns out this whole zippering thing happens in 2 main steps. First, there's a pretty slow step called nucleation. This is where just a few of the right base pairs bump into each other and form a little stable connection. But once that happens, once that initial lock is made, the second step is lightning fast. The rest of the strand just zips right up to form the complete double helix. The initial connection is the hard part. The rest is easy.
So this isn't just some cool trick that nature does. Nope. It's actually one of the most powerful tools we have in the laboratory. So let's see how scientists actually put this principle to work.
The basic recipe in the lab is pretty straightforward. First, you denature the DNA. You basically melt it with heat, splitting it into single strands. Then, you toss in a specially designed single-stranded probe. After that, you cool things down. That's called annealing, which lets your probe find and bind to its target. Finally, you just wash away any extra probes and look for where your labeled probe has stuck. Simple as that. So this probe we mentioned, think of it like a tiny molecular detective. It's a short piece of single-stranded DNA that we've built to be the perfect mirror image of the gene we're hunting for. And to actually see it, we attach a label, kind of like a tiny glowing beacon, which could be radioactive or fluorescent.
But wait a minute, what if you're looking for a sequence that's not a perfect 100% match? Can you still find it? Oh, absolutely. And the way scientists control just how precise this search is by tweaking a little something called stringency. It's actually pretty clever how this works. Under high stringency, think high temperatures, only a perfect, exact match is going to be able to stick together. It's like searching for an identical twin. But if you use low stringency, by lowering the temperature, the probe will be a little more forgiving. It'll tolerate a few mismatches, which lets you find similar sequences, kind of like finding distant cousins.
And that ability, to find both identical twins and distant cousins, that turned out to be a total game changer for tackling one of biology's biggest puzzles, figuring out the tree of life.
The idea behind it is actually super elegant. You take DNA from 2 different species, you melt them both, and then you mix them all together. The more closely related those 2 species are, the better their DNA is going to zip together. And the strength of that bond, which you can measure by seeing how much heat it takes to pull them apart again, gives you a direct readout of how genetically similar they are. And I'm not kidding, this technique completely rewrote the books on evolution. Sibley and Auquist famously redrew the entire family tree for birds using this method. For years, it was the gold standard for telling bacterial species apart, and it helps us figure out the relationships between primates, including ourselves.
In microbiology, it even became this hard and fast rule. For a really long time, if the DNA from 2 different bacteria showed 70% or more similarity in one of these experiments, they were officially called the same species. That was the magic number.
But, science never stands still. As amazing as this classic wet lab method was, it definitely had its problems. So, as always, we pushed forward to find a better, faster, and much more accurate way.
The old school method was tough. It was slow, it was really finicky, and it was easy to make mistakes. One of the biggest headaches was this thing called paralogous interference. That's basically when your probe accidentally sticks to a similar-looking gene inside the same organism, instead of the one from the other species you're trying to compare. It's a case of mistaken identity that could really mess up your results. So what was the solution? Well, scientists basically took the whole thing out of the test tube and dropped it right into the computer. This is digital DNA-DNA hybridization, or DDDH. It uses sequenced genomes to do the entire comparison computationally. It's hybridization, but done completely in silico.
And when you put them side by side, the difference is just night and day. Instead of physically mixing DNA, DDH compares whole genomes on a computer. The algorithms are smart enough to spot and filter out those confusing lookalike genes, which makes it way more accurate. And the best part? It is incredibly fast and totally automated.
So, we've gone from the lab bench to the laptop, but that fundamental challenge of hybridization is still there. That constant hunt for perfect single-base specificity, even when the conditions aren't perfect.
Here's the thing. There's this fundamental physics problem that's always been a bit of a headache. With long DNA strands, the sheer number of correct matches can easily overpower the tiny energy difference of a single mismatch. You can try to force it by working right at the DNA's melting temperature, but that temperature is a moving target. It changes based on all sorts of things, making it a real pain to control.
But now we're getting into an era of designing smarter probes from the ground up. Techniques like toe-hold exchange let scientists build probes that are actually engineered to be a little less stable. This makes them super sensitive, so even a single mismatch is enough to make them fall off. It gives you incredible specificity without having to perfectly dial in that tricky melting temperature.
And that really brings us to today, and to the future. I mean, think about it. Now that we can design probes that can reliably spot a single-letter change in a DNA sequence under all sorts of conditions, what's next? What kind of new, ultra-precise medical diagnostics are we about to see? What kinds of self-assembling DNA nanomachines can we finally build? That's the big question that's driving the next wave of innovation.